This video will talk about the function of T cells, cytotoxic T cells, and B cells, antibody-producing cells, as part of the acquired immune system. I'm first going to talk about cytotoxic or killer T cell development and activation. What we need are cells that will bind to an infected cell and kill it. So here's an infected cell, here's a cell that will bind to it and kill it. Here's the binding. To do that, we need a way for cells to show that they are infected, and they use a surface binding protein, MHC class 1. They need a pocket inside the protein that holds a sample molecule, and there's the pocket. And they need a sample molecule that's from the bacterium or virus inside the cell. The cells that can kill infected cells are cytotoxic T cells, and they need to have a surface receptor with millions or billions of binding pocket shapes. Those binding pockets have to fit the molecules from many specific different bacteria or viruses that might infect an animal. They rearrange this portion by gene rearrangements during development that code specifically for that binding pocket region. In order to get those many different cells, each of which has a different binding pocket, we need stem cells that will undergo gene rearrangements as they divide. So here's the stem cell, which divides to form millions to billions of different T cells. And each one has a different binding pocket shape. In this case, 1A, 1B, and 1C. And we've eliminated all but one of the A's, all but one of the B's, and all but one of the C's. And that will code for a T cell receptor that has a specific shape in its binding pocket. Finally, we need a way for these individual killer T cells to be inactive, doing nothing when there's no infection, but then to replicate many times, but only when their target has invaded the body. Here's the story that I'm going to run through. Stem cells that divide during development to form many different types of T cells, each with a different combination for the binding pocket, and therefore each able to bind to a different target molecule. MHC class 1 proteins that are expressed on normal body cells and that take samples of whatever's inside. In this case, if it's bacterially infected, it takes a part of that, loads it into the binding pocket, moves the whole thing out to the surface where it's presented, and only a B cell that has exactly matching binding pocket to bind that particular target will bind. As part of activation, we need a second event, an antigen-presenting cell that also binds the same fragment, puts it into a related MHC, an MHC class 2, that binds to, again, a binding pocket in a helper T cell, which then releases a second factor, a cytokine, a chemical messenger that says become activated. And so our specific T cell becomes activated only if it gets both of these events. Has to bind and has to receive this signal. Once it does, it goes through many rounds of cell division to make many identical copies. Notice that these are the same A3B1C2, A3B1C2. Now these many different copies of this cell move through the body, and whenever they encounter an infected body cell, they release perforins, which put holes in a cell, and other molecules that kill infected body cells, preventing further development of the bacteria and viruses and exposing those bacteria and viruses for destruction. Let's start with stem cells during development, which have all of the different possible combinations for the binding pocket of a T cell receptor here. During cell division, only a subset are retained, so we have different binding pockets. And therefore each one will bind only a specific target. Now a healthy body cell has MHC class 1 proteins, and it's taking samples of normal self molecules. And any T cell that could bind a self molecule is killed during development, so in early stages of embryonic development. And so these provoke no response. So we get no response from a healthy cell. But a bacterially infected cell has these molecules inside it from some bacterium or virus, and fragments get moved into MHC class 1s that then hold those fragments on the surface. This is an indicator that it's an infected cell. This is not a molecule that should be there normally. Fragments of these wind up in antigen-presenting cells. So a fragment gets moved into an antigen-presenting cell, and then that MHC class 2 with the fragment from a virus or bacterium get presented on the surface. When a helper T cell, which has also gone through gene rearrangements, binds, here's our antigen presenting cell with MHC class 2 and the virus or bacterium, and if our helper T cell has a matching piece, that activates a second messenger cascade, which results in calcium entry and release of a molecule. In this case, it's a cytokine, a signaling molecule. And that specific event indicates that this antigen-presenting cell has a fragment that is a foreign fragment, was recognized by a helper T, so called because it helps the cytotoxic T become active, and it releases a cytokine. If there is a nearby killer T that has bound the same fragment, it has receptors that will bind to that cytokine. Here's our helper T again. It's bound a fragment presented in an MHC class 2. Here's an MHC 1, also holding that fragment. And those two events have resulted in a second messenger cascade from that binding and a different second messenger cascade from this binding. And these events will only happen if there's been an infected cell that activates the T cell and at the same time a helper T. You might wonder why we need two events. This reduces the chance of false alarms. The killer T has to detect its target, and a helper T also has to detect the target. The risk of false alarms is worse than the slowing down the process. Once activated, once these two second messenger cascades have happened, they trigger cell division. And so we get many rounds of replication, potentially to thousands or millions of cells. And after replication, we have many mature killer T cells that are now activated. Notice they have the same set of regions for this binding site, and so they'll bind the same target. So now with these activated killer T cells, they can bind to infected body cells which have put fragments of these invaders, bacteria or viruses, into MHC class 1s, and because they bind and match, that will activate a second messenger cascade. And that second messenger cascade leads to the secretion of perforins, which insert large pores into the cell and cause them to leak and release highly reactive oxygen that winds up killing these cells. At which point, viruses and bacteria no longer can replicate and is now accessible to the rest of the immune system. Toward the end of an infection, another event happens, which is that some of the replicated cells become memory cells. These are inactive and dormant, waiting for the next infection. And in the next infection, rather than just having one or a few cells that have this binding site, we will now have hundreds, thousands, millions, maybe even more of the memory cells, and the reaction to a new infection is much faster.